I'll launch into this session, which is a transition from what we didn't finish last time, and we're finally going to move into immunotherapy after a very long set of background presentations. And we talked last time about newly diagnosed transplant eligible myeloma. And we finished with showing our benchmarks, which before we had RVD induction, when we just had CyBorD, a transplant, and Lin maintenance, our median benefit from our median survival from the transplant was, if standard risk cytogenetics were found, was over 13 years. So that's our starting point. And we are anxious to integrate immunotherapy into the transplant landscape. I didn't get to talk about that much last time. We did have our CMRG trial that gave isatuximab with CyBorD and then with lenalidomide maintenance. That was a high priority CMRG trial, and really the only one that provided that type of antibody outside the rare person who has private insurance to cover it. And that trial finished in 5 minutes. And we will have another one that integrates DARA, but it is one that randomizes to CAR T versus stem cell transplant. But there are provisions for DARA RVD for induction only. Now, you might imagine that this will not be done in every center in Canada, but a bit of good news, there are other ongoing trials to integrate other immunotherapies, such as bispecifics, into the transplant sequence. I thought what I'd do is we'll talk about the immunotherapeutic drugs. We're getting started today, but then we can loop back and it might have more meaning about what immunotherapy trials in transplant eligible patients are available in Canada, so you'll have a better picture of the lay of the land. Now, I realize that's a long introduction, but it sort of closes the loop from last time, because the other area of newly diagnosed patients that we had not touched upon is management of transplant ineligible patients. And I'll start with that and then transition into immunotherapy drugs, and focus on Belantamab, which we've had in Canada for a while. So with that very long-winded introduction, I'll ask Romina to move to the next slide, please. So just again, the goals of this series is to provide a scientific and patient-oriented perspective of myeloma disease and biology, and continually set the stage for an ongoing series. We're moving into the new treatments available, and we are going to be moving in, as we talk about these drugs, try to provide an idea of where you might be able to get them in your province, and under what conditions, and that is a moving target. And then, because there's just one speaker, and we've tried to focus on these two subjects I've just said, hopefully we'll have time for questions, which always seems to get left out at the end in efforts to try to cover a great deal of material. Next slide, please. So this is the master algorithm for managing myeloma in Canada. And again, we talked about transplant eligible, where we were, where we are now, and a little bit about where we're going. And then today we'll focus on transplant ineligible. This is determined by age, or different aspects of frailty. And now there's a gray area of this back to patients who might be considered these days transplant ineligible if they're over 65, despite the fact that for years we tried to transplant as many people as possible because that was always the winner, we thought, in getting the longest first remission. Next slide, please. So I thought I'd start by talking about concepts and then the different regimens within those concepts. And I'll do this historically too. So when myeloma therapy was first started, like almost all other treatment for any cancer, we had an approach that we called fixed duration treatment, when there was a set of drugs put together and then given for so many cycles, and then stop no matter what. That was it. And much of that was because the older drugs we used were quite toxic and could slowly injure the normal bone marrow. And that was satisfactory for diseases, particularly certain lymphomas, certain leukemias. That's all you needed. And then things changed later in the 2000s for myeloma in particular, into and based on the introduction of drugs that are different from classic chemotherapy, which I think of as a poison for cancer cells. There are different drugs that don't poison the cancer cells, they inhibit them, they suppress them. So they are designed to get the best effect if you give them continuously. And some of you have heard, I often use the concept or the analogy that it's like blood pressure medicine. As long as you take it, your blood pressure is controlled. If one stops it, blood pressure will go back up. And this description of continuous treatment really is useful for drugs that manipulate the immune system. They're not classic cancer cell poisons. And our drugs in myeloma that were used in this manner were the immunomodulatory derivatives, the IMiDs, so thalidomide, lenalidomide, and pomalidomide. So there are studies that have given thalidomide or lenalidomide for fixed duration, so many cycles, and then stopped. And that is, in a way, counterintuitive to how these drugs work. But why has that been done? Well, one of the drivers has been cost. Before lenalidomide went generic, the list price, now that's not always what the province paid, but it was up to $1 million a year. And, that is a massive burden on any public healthcare system. It is generic and has come down, but there's great interest in trying to optimize how long it's given to get benefit, but not break the healthcare system. And we hear consistently and understandably that all cancer drugs carry side effects, and treatment breaks are certainly a high priority for our patients. And in particular, if you're asking your patient to stay on a drug indefinitely, you have to stay on top of the side effects, or quality of life can be impaired. And the goal of treatment is to control the disease as long as possible with a good quality of life. And to maintain quality of life is an ongoing effort that is all too easy to get lost in a brief clinic visit. Next slide, please. So if we go back to this master plan, one can see that transplant ineligible patients now have 1, 2, 3, 4, 5 choices that are funded, and they're a mix. Most are no longer fixed duration, but are given continuously until progression. Back to that continuous type of treatment. Next slide, please. I'm going to go through the history of this, and some of you may be on these regimens, may have had them before, but to give you a historical idea, and I pair this with my experience in Toronto, which started in 2001, but all we had if patients couldn't go to a transplant was good old oral melphalan and prednisone. And in Canada, we tended to give cyclophosphamide and prednisone. And I wish we were all in one room, because it would be interesting to ask people to raise their hand if they had ever had old M&P, but that's all there was for 25 years. Then we were able to, in a trial first done in Canada, is create Cybor prednisone. Then now we use dexamethasone by adding bortezomib or Velcade. Then we had this new drug, lenalidomide, with steroids, RD. Then bortezomib was brought into that as a triplet, and now we've added the monoclonal antibody daratumumab to both RD and Cybor-D. And I'm going to hone in on this a little bit more to show you benchmark outcomes, realizing that these median values don't define any one person's course, but sort of how a group of people do. Half people go longer, half people may go shorter. Next slide, please. So melphalan and prednisone is very easy to give. It's 4 days a pill about every 6 weeks for up to 1 year. Minimal side effects for an experience, but only about half of patients had a partial remission, which was usually enough to knock down symptoms, but was nothing like a deep remission, complete remission, very good partial remission, and it didn't work over a year. And the problem with melphalan, which now has been relegated just to one high dose before a stem cell transplant, but if you give it every 6 weeks, it damages the bone marrow over time, so that at the end of a year, the platelets may not go over 95 and the neutrophil count may be running 1,000. Well, that's compromising the marrow a bit, and we are hoping our patient's going to receive a lot more treatment and their marrow is not as healthy. So actually, Canada was very wise in the 80s, started doing trials with a different related drug to melphalan called cyclophosphamide, which can be given weekly, not necessarily fixed duration. And we know now you can give 1 pill every other day instead of 8 or 10 pills once a week. So this actually was very helpful for patients in protecting the bone marrow. And then, as I said, we added bortezomib or Velcade for Cybor-D. And one of the things we did in Canada, in a consistent effort to try to Canadianize our regimens with less toxicity, preserving the benefit, is insisted with our sponsor to give bortezomib weekly at a time when the whole world was giving it twice weekly and seeing really problematic peripheral neuropathy, painful neuropathy. And when this regimen was studied in newly diagnosed patients, 85% of patients responded after only 9 cycles, which is all the province would fund. The benefit was over double melphalan and prednisone or cyclophosphamide and prednisone. And the weekly bortezomib, it's not free of neuropathy, but our incidence in Canada is markedly lower than other countries, which particularly in Europe and the US, they continue to give it twice weekly because that's what the package insert says. And I think my own opinion causes a lot of unnecessary misery when we've shown you can do it less frequently, same benefit, but it wasn't done in a phase 3, huge multi-million-dollar study. One of the attractive aspects of good old Cyborg D is, in addition to less neuropathy, it is very easy to give to people who are just diagnosed with myeloma when they may have kidney damage or their blood counts are low because myeloma is filling up the marrow, and one can titrate it and clean out the marrow, help the kidneys, and move on. And that's still one of its potential benefits. But then sort of around, I think the paper was published in 2012, lenalidomide was moved from the relapse setting to newly diagnosed patients. It's given once a day for 3 weeks, and then dexamethasone 1 day a week until progression. And this had about the same response rate as Cyborg D, but it has been shown even in our database trials with our real world experience in Canada, last updated by our Canadian CMRG member, Rayanne Cadby, to give almost another year median. And when I closed my practice, I had some people still on RD from when they got it on one of the trials, 8+ years from starting. So it can really be beneficial. The downside is it's continuous, and it's been shown in a trial that only gave it 18 months that your first remission lasts longer if you stay on it. Now, again, there were important learnings. In Canada, if you line up hematologists, none of us start anymore lenalidomide in someone who is not going to a transplant with the dose in the package insert, which was 25 milligrams a day. It's just too hard on people. There is fatigue, diarrhea, and sometimes the blood counts go low. And I think all of us practicing now in Canada say, okay, that's what the trial did. And then, yes, you can stop for side effects. They'll go away. Then you reduce the dose and people are better. But why do that if you know that's going to happen in a lot of people? So we have evolved that we don't start at 25 milligrams. You usually don't need to. You're going to have to dose reduce anyway. And I would put forth that you don't want to lose the heart and mind of someone who's being asked to take these drugs continuously by making them sick the first month or two. So we all start maybe 10 milligrams a day for 3 weeks, maybe 15 at most. No one starts at 25. And there's a lot of need to attend to the frequent side effects. Fatigue is less with the lower dose. And diarrhea can usually be managed by either with a bile acid binder, either Lodalis or cholestyramine, which also needs to be titrated, because if you give it every day, people may get too constipated. Maybe it's 3 times a week. You have to work with your patient to get this tolerable, and be ready to dose reduce, because I think with these newer drugs, it's not the maximally tolerated dose that you can give in a clinical trial before people get too sick to go higher. It's, as you'll see in another slide, I think in myeloma, and the FDA is finally accepting this, it's the minimal dose that works, that people can stay on and have a decent quality of life. Because the approach is, we're not curing the disease with this, but we have a drug that can work years, and let's make it as tolerable as possible. Around 2018, you can see this theme here. We were funded to add bortezomib to RD. And again, in Canada, there's several ways to give it. We give weekly bortezomib, and we give lower dose lenalidomide than is published in a big US study. We give 8 cycles and then drop it down to just len. We don't necessarily call it maintenance because it's suppressive therapy, sometimes with steroids, and maybe try to reduce the steroids. And now we're pushing the benefit consistently over 3 years to 3.5 years. Whereas if you look at how it's given in one of the huge US trials, the incidence of peripheral neuropathy was tremendous, including very serious peripheral neuropathy. And it's again, my bias, it's unnecessary. And we follow through our CMRG database that our benchmark results are just as good as giving it at maximally tolerated dose and then having to dose reduce. So we are continually, I mean, I think if we're going to adjust doses of a regimen that's been reported in a trial, we have to be sure that we're still getting the same effect. And I think that's one of the benefits of the database, is that we are showing our benchmarks are perfectly comparable in this situation with the more toxic regimens. Next slide, please. A few other thoughts about continuous treatment, because it is undoubtedly challenging. And, you know, I'm always pointing out differences between US patients, even though I haven't practiced there in well over 2 decades, tend to want to be on things, fix it. And Canadian patients are, I don't want to take it unless I absolutely have to. It's an interesting way of approaching health. But it's hard to have to think about staying on medication long term. So ideally, we'd want to know how long we must treat each person to balance risk versus benefit. And that remains elusive because of the variability in the disease, in the biology, and a biology that changes over time, as we've discussed in previous situations. And I've always found it hard to give up a treatment that is working. I'm perfectly happy to be sure the doses are tolerable. That's the other side of the coin. But if we stop it when it's working well, funding may or may not allow us to use it again. That has loosened up a bit in recent years, but it used to be you get it once, particularly in Ontario, and that's it. But even if we can use it again, observations is since we're not curing the disease and it will come back, we don't know how well restarting what we stopped will work. And it's uncertain. It could work, but often it doesn't, because the myeloma has grown new subclones, let's say, that are now resistant to that agent. Or sometimes the myeloma, which had been grumbly at the beginning, comes back in a very angry way. And if we've optimized our continuous treatment where someone is on a very low dose of Revlimid and a low dose of steroids, it's not always the case that just starting those low doses again is enough. You have to go back up at the top and then add a drug or two. So it may be the treatment burden is, in my experience, usually worse. So it's a balancing act. And again, some people may do very well for months, occasionally years off therapy, but we we're working on it, but we don't have the tools yet to predict who can stop and the disease doesn't start drifting up fairly quickly, and even better, can be retreated with the same thing. And we know with general studies, remissions tend to get shorter with each line of treatment. Now, that too may be changing as we get some of the newer immunotherapies, mainly CAR T, but that's still a ways off. And um, until then, we prove that, and CAR T is going to be moved up earlier and earlier. So I think with our continuous treatments, they must be tolerable. Dose adjustments are absolutely key. One has to keep up with monitoring not only of blood counts, but how our patients feel and be ready to adjust, and to look at those issues that make our lives enriched. It's not just diarrhea, but how many clinic visits? Can people travel and visit family and have some recreation? How can we preserve energy? And all these aspects that are so important, but don't make it into clinical trial assessments. Yeah, we measure quality of life, but there are other aspects that um, go into that equation. Next slide, please. So we're very excited that over the last 2 years, we finally have funding for daratumumab with RD until progression. Now, we also, at the same time, got funding for adding daratumumab to Cybor-D. There's only 9 cycles of Cybor-D. That part's fixed duration, but the DARA continues. And there were 2 big trials. Maya was the DRD trial versus just RD. And the overall response rate was 93%, and the median benefit was about 5 years, which is the best ever. And the results are in the same ballpark as the results of stem cell transplant in terms of the benefit of the first remission, a stem cell transplant without immunotherapy. Now, once immunotherapy is routine, or for those individuals that have been able to access a trial or been able to access immunotherapy, it's going to be longer with immunotherapy. And also, when we now can use RVD induction, like we talked about with transplant, that median benefit looks like it's going to be closer to 6 years. So the transplant's still coming out now ahead with RVD induction, and we'll take another quantum leap once we can add daratumumab to that. But 5 years was so much better than 9 months, 10 months with M&P when I first came. And we are now in the process of designing, planning, and conducting database analyses using our CMRG database now that we have 2 years of treatment, of looking at our early results in several different observational studies that different hematologists across the country are working on. So we're anxious to see how our Canadian population is doing. So I think it's not a finished product, but the impact of immunotherapy in myeloma has really been quite profound. Next slide, please. So now we're after 6 sessions, finally moving in more to immunotherapy, which is such a hot topic. And the segue is we've talked about how daratumumab helps RD. We've talked about, hinted at how it helps with stem cell transplant. And it's the first monoclonal antibody that's been funded in Canada. It was initially funded, as some of you know, for relapse myeloma with those same partners we've talked about now at diagnosis with bortezomib or lenalidomide. And now it's available at diagnosis for patients who are at least age 65 and transplant ineligible. And another database project is going to look how having this available for 2 years has affected the decision to go to transplant. Are we seeing more people that are choosing with their physician not to have a stem cell transplant? Now, it's not considered an option later, but they'll just go with DARA-RD because it avoids the unpleasantness and hassle of a stem cell transplant. And part of that decision is after a stem cell transplant, one is on one drug, len maintenance, usually, and with DARA-RD, those 3 drugs are continued until progression. So what's the preference and the feeling about treatment burden? Is it, bomb it out up front and stay on low dose len, or is it be on 3 drugs indefinitely without a lot of data to alter that at this point? But that's a discussion people should have with their physician. So we haven't done this before, but I think it might be a good time to take questions about first line treatment in transplant ineligible, because then we're going to veer off and talk about immunotherapy and some of the choice in older individuals might be lost after we talk about some of this newer stuff. But so I'm happy to take your questions or comments at this point, and then we'll launch in to immunotherapy.
I have a quick question. How do you determine if someone is over 72, 73 and is fit? Are they still not eligible for the transplant?
That comes up frequent, or used to come up all the time, and there isn't a consensus across the country, but I think most centers, and certainly Princess Margaret, has considered the vague term of physiologic age. What's your body's age? Your numerical age. And if someone has really good fitness, is a marathon runner, or very fit, our center has done people up to 73, 74. Again, this is my own experience. And what I have found as a counterbalance to trying to push it to the max is and again, it's hard to prove this scientifically, but what I would tell my patients that we're having this discussion is if people are, have good heart, lungs, kidneys, they'll have very much the similar outcome of a stem cell transplant as someone that's a few years younger. But again, this is personal observation. Even if with fitness, the recovery time for that fatigue, really profound fatigue in the first weeks after transplant, can be much longer. And I told it when I was in the US, which had no age limit, may have been about 76. And this individual literally at a year was still absolutely exhausted. And, so I think that's part of it. There's just a sense that even though we're fit, our body doesn't heal as fast. And maybe we decondition faster during the time one can't run marathons during the weeks of a transplant. So I worry more not about the transplant's going to have fatal side effects, but how long a recovery of that energy and stamina level is acceptable when there's another alternative. And that's really what I worry about more than anything else. I hope that answers your question.
Yes, thank you.
We do try not to put a cap on it, but I really am reserved after about age 74. I don't recommend it just because of that risk. And who wants to be on the couch for 6, 7 months? Any other questions? No?
I guess no.
Okay. So moving on to immunotherapy. I think a little bit of background because this will frame our next few sessions as we really dig into the different drugs, how to access them, what they're used for, what are our expected results, what are the challenges. Is there two basic types. One is monoclonal antibodies. That's a drug treatment. It just happens to be an antibody. This type of immunotherapy is made in a lab and generates an antibody that sticks on myeloma cells. In general, it's given IV, although most of them are now being formulated under the skin, subcutaneous, which is wonderful. No IV. And there may be a loading dose to get the levels up like once a week for a month or 2 months. And then once the levels of this antibody are high enough, you can back off to every 2 to 4 weeks, which gives our patients a lot more continuous treatment because antibodies break down in our body. And I'll talk about their mechanism of action in a minute, kill the disease. So the other big type is what we call cellular therapy. We're actually giving intravenously cells. It's very much like a stem cell transplant, but the cells we give back are different. They're not the seed stem cells to plant a new bone marrow, neutrophils, hemoglobin, platelets. Immune cells, a type of immune cell called a T lymphocyte, which is circulating in your blood. We just don't ever look at it or talk about it much. And those T cells have been reprogrammed in the lab to hone in and attack myeloma cells and kill them. These are given intravenously. They're frozen and thawed and given in intravenously, just like stem cells. And it is designed to be a one-time treatment. Although stubborn myeloma is, there are pairing it with some or a different therapy to keep this disease from waking up again. And it is a one treatment. It's lymphoma, just like fixed duration regular chemo can cure a lot of myeloma, lymphoma, but myeloma is a different disease. So right now, when we get funding as we anticipate, it one-time treatment. Next slide, please. So a little bit about antibodies, and many of you on this in this meeting have heard about that plasma cell. Each plasma cell makes a unique antibody, and it's that Y-shaped molecule. The double end, they each recognize the same target, which I've called it an invader. It can be a virus, it can be a bacteria, it can be a part of those, a tumor cell. And it completely have the ability to kill the target, but it raises a flag of immune cells to come in and help finish the kill. So it has damaged a cancer, to damage the invader, but it's even better if it can calm the troops. So in a patient, cells in bone marrow are because with these two, they all make an antibody. It doesn't known invader, and we use it. Many years ago, they learned that you could take plasma cells in a lab and turn them into little factories to make a monoclonal antibody, and you could design them to make that antibody stick on a particular invader outside. And this technology is not new, and so you can do this very efficiently and very easily. You don't have to worry about that blue diagram, but it shows that you know how you design these little factories in the lab to manufacture a certain monoclonal antibody. And the key was finding markers on the surface of a myeloma cell that were mainly only on the myeloma cell and weren't on lung, heart, other cells, because you don't want to give these monoclonal antibodies back, have them home in myeloma cells, but have all this collateral damage on vital organs. So it took a while to find unique markers on a myeloma cell that you could make these lab antibodies stick onto without hurting a person. So that's been done now. Next slide, please. And this is a schematic. That big blue cell with the purple nucleus is a myeloma cell. And all cells have all sorts of markers on the surface. And some of these are on all our body cells. You don't want a monoclonal antibody against a marker that's on all of our body cells. The antibodies would attack our whole body. But you can see there are several of these, and I've simplified this to the most common 4 surface markers that monoclonal antibodies have been created for. And the marker in yellow is called CD38, and daratumumab and a related drug from a different company called isatuximab, and I've shown the brand names Darzalex and Sarclisa, are made that when they're given IV, they home on this marker on the myeloma cells and start to kill it and mark it for destruction so other immune cells are brought in. And this is the concept behind antibody type of immunotherapy. And these are called, it's kind of a funny term, naked antibodies. They really are very similar to one's own antibodies that are being made against COVID or other bugs, but they do their job like other antibodies. Next slide, please. But the technology for this is really moving at warp speed. So antibodies, monoclonal antibody therapy is really easy to give compared to the kinds of side effects we see with cancer drugs in general, the poisons, etc., in that they act like our immune system and just focus it on a one type of bad cell. And the side effects have to do with antibodies or proteins. And when we inject proteins in anyone, we can get fever and chills. We don't get hair loss. Nausea and vomiting is rare. It doesn't go with these three. We can sometimes get rashes or allergic problems. And now with the pre-medications that are routinely given at the beginning of these monoclonal antibodies, these are uncommon. So they tend to be very easy for people to take and much more appealing than poisons, I would put forth. And then with these pre-medications that are usually antihistamines or one's an anti-asthma drug, Montelukast really helped. It's rare to have a marked reaction anymore, and giving daratumumab subcutaneously under the skin, they're almost gone. Isatuximab is given, and you can give them faster IV than we knew at the beginning. And isatuximab infusion, we're learning, and this will come out in one of our CMRG studies, you can even give it appears even shorter than the hour or so. And the risk of these sorts of protein reactions go down after the first couple of doses. So in the long term, if one is getting these every month, or it's very rare to have a side effect from that infusion. The antibodies are really good when you combine them with other treatments. The naked antibodies, though, by themselves, because it's all immunotherapy, are not as potent as we would like. So the sophisticated steps to enhance them and make them more powerful are twofold. One is like this cartoon, is they've taken that Y-shaped antibody and stuck a poison on it. It's shown here as the red cytotoxic drug. So then when you give these intravenously, and we're going to talk about Belantamab, this is what it does. You give your antibody IV that goes and recognizes BCMA on the surface of myeloma cells, but then it also, and that calls in the forces to help kill the cell, but then you have a toxin against that cell there to enhance cell kill. And these are called ADCs, antibody drug conjugates. And they're being studied in a wide variety of cancers. Of course, the antibody doesn't stick on BCMA, it will stick on a lung cancer cell marker or a lymphoma marker, but they're coming into development because, as I said, monoclonal antibodies are easy to make once you identify a target on a cancer cell. So I'm going to move to talk about that in the last few slides, but the other one that we'll get into much more heavy duty next time are the BiTEs. And BiTEs is a nickname for a long title that's called the bispecific T-cell engager antibody. So we like to say BiTE. Bispecific means it recognizes two targets. And T-cell engager means one target is the killer T-cell, and the other target happens to be a plasma cell. And if you can see the diagram, most antibodies, that upper two lines of the Y, stick on the same marker. But this is ingenious. One sticks on the cancer cell, but the other sticks on the killer cell. And so it brings them side by side. And the T cell then is right there. It doesn't have to circulate in the bloodstream and be called in by the antibody. It is right there. And the two abut each other, and the T cell directly kills the myeloma cell. And the way it does it is release these special cell killing substances that don't hurt anything else, that are shown in those little dots, porphyrin and granzymes, and really cool stuff that our body uses to get rid of unwanted invaders. Now, there's a war here between these cells, and when there's a war, there's smoke and fire. And those are some of the side effects that we'll talk about in later sessions that make giving bites such a specialized area with special considerations along with their very cool mechanism of action. So maybe again, I think for once we're doing okay with time, but maybe I'll stop again and see if people have any questions about these. We're really getting into the meat of immunology, but hopefully it gives you a visual picture of what these drugs do when we then talk about names and side effects. So I'm happy to entertain any broad questions the best I can. My goal is it sort of makes sense. Any questions?
Guess not.
No, I need Suzanne here because then she'll say, well, Donna, that's all very well and good, but what does it mean for me? But anyway, okay, we're now going to talk about the ADC that we have had in Canada for a while and that Canada's done work with, and just some aspects of Belantamab, which aside from daratumumab and isatuximab has been the immunotherapy we've had access to until the very recent past. Next slide, please. So in my effort to have a visual, this is our myeloma cell with the cell surface markers, and the target for Belantamab is different, obviously, than for daratumumab, but it's this BCMA orange figure that's sticking out of the cells. And the Belantamab is the orange antibody, and I've just put a bomb next to it because it does have more potent killing power. Just giving a plain old naked BCMA monoclonal antibody without the bomb attached to it, or it being in the form of a bite, isn't potent enough. So we've had to move beyond just giving an antibody. Next slide, please. So Belantamab has a certain toxin attached to it, and we've actually had access to it through trials for quite a few years. And again, it was initially developed to give the maximally tolerated dose in trials. They give as much as they can before side effects are too much, and then they further study the dose level below the maximally tolerated dose. So it's that give as much as you can that can be tolerated idea. And it was initially studied at this certain dose that I showed here, 2.5 to 3.4 milligrams per kilogram every 3 weeks. And I actually had patients well before COVID who did quite well, but its main side effect of all things was to damage the cornea of the eye. And I describe it, the cornea is sort of like the windshield over your lens. Um, and if it's damaged, it makes the eyes feel dry or gritty. Vision can be blurred, and the visual acuity may not be as good. Now, the good thing is when you hold the drug, it completely reverses. But that's an annoying toxicity, and it's actually not due to the antibody itself, it's due to the toxin attached to it. It can decrease the platelets a little bit. We'll be talking more, but these antibodies that we give, be it daratumumab or belamaf, or certainly the bites in the future, they stick on plasma cells and they stick on myeloma cells, but they also stick on normal plasma cells. So those are destroyed as well. So good plasma cells are part of our immune system that fights infection. So all these antibodies can carry an increased risk of other infections because they're inhibiting part of our many different platoons of our immune system. And we have approaches to counteract it. It's very important that certain preventative antibiotics be given always, and that keeps those infections away. That it's increasingly appreciated that, particularly with the newer ones, we need to be sure our vaccinations are up to date, aside from COVID, because they don't take as well if these drugs are suppressing our normal plasma cells that make the antibodies to COVID and stuff. But that's all being worked out. But by and large, except for this eye side effects, the belamaf or belantamab mafodotin really was a very new important side effect. And we had a trial at Princess Margaret probably 6 or 7 years ago that was quite good, even though it was at higher doses. That trial closed, and then for a number of years, I'm sorry I didn't double check the date, but well before COVID, there was what's called a special access program through Health Canada to get belamaf one patient at a time. And I'm mentioning this because SAPs, or special access programs, have been an important way to access drugs before they're paid for publicly or even available through private insurance. And an SAP requires that the drug company that is studying the drug, that is looking promising, goes to Health Canada and says, we want to open up an SAP. We're going to give the drug for free. Health Canada has them do their side of the paperwork, and then there's sort of a clearinghouse. And the way it works is for each person that we want to get this drug that we can't buy anywhere in the country, we have to write to Health Canada with anonymous information about our patient. And the catch is, and they want to help, but you have to indicate that one's patient has had every other drug approved for myeloma before they'll release this drug. And they can't check your records, but you have to indicate your patient has had everything, and lack of funding didn't count. But anyway, it is a mechanism to get this, and then it's given to your hospital's pharmacy with instructions on how to give it, and then it has to be reordered every couple of cycles. And it is a limited, but it is a way to get drugs when you absolutely have to. There's a fair amount of paperwork, but it is doable. And Belantamab has been available through an ASCT. But next slide. We through CMRG and Suzanne Trudell, who'd done some initial legwork with Belantamab, as the PI, we wrote what we call the Algonquin trial, because all our CMRG trials are named after a geographic area or a national or provincial park. And since this one originated in Ontario, we had the Laurentians trial with Palm and Dara, and we've had the Mewasan trial from Saskatchewan with rapid infusion Dara. So this is the Algonquin trial. And what we set out to do is, okay, Belantamab by itself can be quite effective, but combinations always turn out to be more effective. And we wanted to combine Belantamab
Why does my thing freeze?
I think she got disconnected. Let me check. Okay, something happened with her connection. She'll be back soon. Okay.
I apologize. I have the world's worst internet, despite anyway, maybe people could take a bit of break. But we had the flexibility, fortunately, to try to come up with the best tolerated effective regimen, and I hope made a contribution in many respects that it's not 3.4 milligrams per kilogram every 3 weeks. The sweet spot appeared to be a lower dose every month, which is also convenient for our patients because they're not coming in every 3 or 4 weeks, they're coming in every 2 months. And this was the initial portion of the trial was just published earlier this year in Nature Medicine. Next slide, please. So, Algonquin, oh no, is now been treated over 100 people, and it's still ongoing. And I won't go a lot into the details, but there's some terminology that will be using. Health Canada is very, and the FDA, very focused on how many different regimens people have had before a trial. And, you know, induction with or without a transplant is line 1. Then there's line 2 for first relapse, line 3. And to get these new, in most time, people have had to have 3 prior lines. And the median in this trial was 3 prior lines. The other big terminology we use is these new immunotherapy position for people who've had at least 3 lines of, and somewhere in that treatment has been included Velcade, Revlimid, and daratumumab, or variations. So they've had one drug of the 3 main classes: a protein inhibitor, an IMiD, and a CD38 antibody. And exposure to all our new fancy immune drugs. So half of patients it's over. Even just still respond. I'm sorry, there's a typo here. We're not seeing any new. We're still dealing with the eye toxicity. People very rarely have to stop the drug. We have to skip a dose and let the eye problems resolve. And we're exploring other ways to address the eyes. One of the approaches that Martha Lozada in London has developed and was one contact treatment in the not, and we're seeing they protect when the cornea is damaged. We think what's happening is, for some reason, the warhead on Belantamab, our cornea apparently regrows. It's like the windshield of your car gets a few scratches and dents, and then, you know, it regenerates itself and your windshield is clear again. And so if that regrowth is slowed down by Belantamab, you know, it's going to come back, but you have to give a break, and then that windshield grows back and you don't have dry, gritty eyes again. So it's been well shown this is not permanent, but it's annoying. So we're trying different things to make this as tolerable as possible, because the other side effects are not that I don't want to sound judgmental, not that bad compared to bad blood counts. Even infections aren't as problematic as with the bites, as we'll talk about. So, I mean, there's room for improvement. But what's also rather remarkable, I mean, I review all the side effects for these CMRG trials. I'm finding updates, people have been on it since 2001, which for fourth line treatment many times is pretty darn good. So we want to make it more tolerable. And there's so many new drugs coming, which is great, but we got to get them to Canada. And we've all seen that can be agonizingly slow. And I will say this regimen has given Canadian patients access to a decent immunotherapy whose benefit in these patients is not much different from a single agent bite in this session since January for this particular trial, 2019. So, we do have a lack of things sometimes, but I just wanted everyone to know that we're working very hard at CMRG to find crevices we can get in and bring new drugs to patients. That's really our main goal, is getting drugs to patients. So this is a bit of a story. And we are continuing the trial, and we'll have some bispecific trials, and they're all focused on how to suppress the disease as long as possible with a good quality of life and not giving everything at the maximum tolerated dose. That's a different strategy, and I think all of us in our group feel that that's antiquated and not the best way to give immunotherapy. So stay tuned. I think this is the last slide. Do you mind going on? Oh, yeah. So stay tuned. We're going to be talking more about the bites, how they're available, where they're available, what we have coming up on the horizon. And we'll be talking more about the BCMA bites. It's interesting. There are a whole bunch of them. Almost every drug company has their bite. I mean, hopefully competition will bring the price down, which is priced about as much as a CAR T for subcutaneous antibodies given periodically. So maybe if there's competition, the price will go down. But there are also very promising bites against the purple guy, GPRC5D, and the green guy, FCRH5, that we've had at Princess Margaret at various times for the last few years. So, the studies now are combining bites or combining bites with daratumumab or other drugs, or using them sequentially. So there's incredible amount, a wave of treatments that are being given with. I don't know if they'll ever completely replace our older drugs, but the future is incredibly exciting, and we're committed to getting as much of these into Canada in ways that are very as tolerable as possible. So I think that is my last slide, and hopefully a bit of a teaser for our next sessions as we get into the real nitty-gritty of what's coming in myeloma. So I think we do have some time for questions, so I certainly welcome them, and I'm happy to go over anything again, because I hope this is a foundation, because we're going to be talking about some really exciting new treatments that are right on the cusp. So thank you very much for taking time out, and also let us know how this time works or doesn't work. Send your questions in to Ramina, follow up on our newsletter, and Suzanne will be back next month to assist. So thank you very much.
So I do have a question.
Sure.
Is there work undergoing to develop I don't know what you would call it, that would attack more than one, like it might attack this CD38 and BCMA at the same time? Would that be more effective, more toxic, that type of thing?
It's a good question. What people are, yes, but they're doing it with giving two antibodies together. There are a number of trials that are given like a bite,
Oh, myeloma talk from
which
Donna
will
Reeds.
which will get BCMA and giving it the same day with daratumumab that will get CD38. So they haven't made an antibody that's reached very far in clinical trial that gets one antibody, both BCMA and CD38, but you're onto something. They are making, we've talked about bispecific antibodies. There are trispecific antibodies that get two of those markers plus the killer T cell, or they're they're exploring, although it hasn't been as promising. They just decided to drop one of the drugs we were studying that doesn't use a T lymphocyte. It uses what's called an NK or natural killer cell. So they're again, once you get the targets, these antibodies are pretty straightforward to make. So they're making tri-specific and all sorts of combinations. What's the low-hanging fruit is giving two antibodies together.
Thank you.
I have a question, Dr. Reese.
Sure.
It's Geraldine. First of all, thank you for your time and commitment. I know you're retired, and I look forward to receiving the information that you provide for us as patients. And so my question is, how can we as patients help to get these drugs to Canada?
Make noise.
Who do we make noise to?
It's hard. It's very firewalled. I think, the feedback on the CADIT forms, I think keeping an awareness of myeloma is a huge problem. And I think it's a difficult climate now, but I think just spreading awareness about the disease. All cancers are experiencing this because one of the challenges is the list price of the bispecifics. Now, this is list. It's not what they're going to pay, but they're priced at almost $500,000 Canadian per year. And you can envision right now our patients getting 2 or 3 of these. So, and it's not just myeloma, lymphoma, et cetera. So the cost is a huge problem. It's interesting that Europe used to, whenever they didn't have CADIT, except for a couple of countries, that does the cost effectiveness recommendation. Should this be funded for what the results are? Europe, once they approved the drug, it was automatically covered by the public healthcare system. That's going to stop next year. They can't afford it. So it's interesting. The Europeans are looking to the UK, NICE, was their CADIT, our CADIT, and the Australian, they call it Health Technology Assessment Group, to try to figure out how do they decide what to fund. Our challenge is mainly financial. I mean, the drugs are good, but I think our fight is financial. And what we are told, and again, don't quote me because I haven't gone to the source data, but for the market for these drugs, 80%'s in the US. So, how much do they need? Switzerland, Canada, and Japan, if 80%, but it won't last in the US. So I think there are efforts to bring different public healthcare systems together. I mean, because the problem's the cost. But I think awareness is a big deal. Respond to whatever surveys, and insist that the results be so good that it's worth the money. That's pretty generic, but I find we're more and more in CMRG into advocacy. It's exciting. CADTH has opened the door now for real world data to help make their funding decisions. And we're there. We've already been there showing our results for where we have gaps, and now they're formally inviting that. So that was one when we first went to them about 7 or 8 years ago and said, we're going to have real world data. We're going to have pilot studies that don't have control. We want you to look at that data. And I'm exaggerating. They kind of rolled their eyes. Now they're looking at it. So our CADTH realizes that they have to broaden how they look at things. I mean, you know, my bugaboo for years was they'll only approve how these drug regimens are given in the phase 3 trials that never get the doses right. And we're supposed to give toxic doses because the trial did it and they got it wrong. And now they're looking at our CMRG that says, look,